Hello and good afternoon. My name is Yohannes Ghebre and I'm an associate professor at Baylor College of Medicine. Today, I would like to talk to you about the potential of repurposing the FDA-approved anti-acid drug esomeprazole for the treatment of morphea. First thing first, what is morphea? Morphea, also known as localized scleroderma, is a rare autoimmune disease characterized by excessive inflammation and abnormal deposition of collagen in the skin and underlying soft tissue. As a result, it causes pigment change, as you can appreciate in the image. In its severe form, morphea can also cause impaired mobility, including joint contractures, as shown in the lower panel. How common is morphea? Currently, the disease has an incidence of up to 3 cases per 100,000, and it is estimated that there are about 30,000 patients in the US alone. It is important to note that women are disproportionately affected by morphea at 2.6 to 1 ratio compared to men. Although the cause of morphea is largely unknown, new onset of the disease in the settings of radiation therapy has notably increased over the years. The image shown provides an example of radiation morphea. So, what is the current treatment for morphea? It is unfortunate that there is no curative pharmacotherapy for morphea. However, immunosuppressive drugs are used to manage the disease and alleviate symptoms. Unfortunately, the use of those agents is associated with significant side effects, including toxicity and risk of infection. It is intriguing to note that none of the current treatments for morphea target fibrosis. This is despite the fact that pro-fibrotic pathways are upregulated in morphea at preclinical and clinical settings. Sadly, there are not very many candidate drugs on the horizon. Of the very few that are in clinical trials, most of them are immunosuppressive drugs targeting inflammation, and none of them targets fibrosis. Well, what if I tell you that there is a generic drug widely available in the market that can simultaneously target both inflammation and fibrosis? Please meet the FDA-approved anti-acid drug esomeprazole, also known as Nexium. Esomeprazole was identified to have anti-inflammatory and anti-fibrotic activities upon high-throughput screening of 130,000 small molecules against an enzyme known to drive inflammation and tissue fibrosis. We then formulated esomeprazole into a topical product, which we named dermaprazole, and assessed its anti-inflammatory and anti-fibrotic potential in a mouse model of radiation-induced dermal inflammation and fibrosis. As the data shows, dermaprazole is a very effective anti-inflammatory and anti-fibrotic product. This is shown by the accelerated healing of the irradiated area in the animals that received dermaprazole in a prophylactic or therapeutic manner, as shown in the last 3 data sets. From the data, you can also appreciate that the corticosteroid-treated animals still show necrotic skin. I don't have the time to show, but we also have positive data in an animal model of localized scleroderma, where the dermal inflammation is chemically induced by bleomycin. So, what are we proposing to do? We're proposing to conduct a phase 1/2A clinical study that is primarily focused on safety and tolerability of dermaprazole in patients with morphea. We are using the standard 3+3 study design to first conduct patch testing in healthy volunteers to assess the safety of 1% and 2% dermaprazole over the course of 6-week treatment, followed by 4 weeks of follow-up. If found to be safe, we will then proceed to patch testing in an unaffected area in morphea patients using the maximum tolerated dose identified in the above studies. If found to be safe and well tolerated, we will then enroll 12 patients with morphea to conduct a 3-month safety and preliminary efficacy study. Here are our endpoints. For safety, we are looking at burning, itching, tenderness, rash, and/or allergic reactions to dermaprazole. For the efficacy study, we will use 2 well-validated tools to assess the disease activity and quality of life. One may ask, what would the proposed study cost? We are asking for up to $70,000 in funding. Most of this funding will go towards study personnel drug purchase and formulation of the product for 30 or more study subjects, including the patch testing. Remember, the drug is only available in oral and IV forms in the market, and it needs to be formulated for this study. Who is involved in the study? I am translational researcher and study PI. My lab has pioneered the dermaprazole technology. Dr. Jacobi is a co-investigator. She's professor of dermatology and exclusively sees morphea patients. Dr. Mayas is a project advisor. She's internationally recognized expert in scleroderma research and clinical care. Dr. Ramamurthy is a co-investigator. She's an expert bioinformatician and biostatistician. She has several years of experience in data coordination for clinical trials. To summarize, morphea is a rare and understudied disease that affects humans across lifespan. It is lamentable to know that there is no single pharmacotherapy that can treat morphea. That's why you have the opportunity to support the first in-human clinical trial to repurpose a widely available generic drug for the treatment of morphea. Supporting this study presents an unparalleled opportunity to leave a lasting legacy in the lives of morphea patients, their families, and the community at large. With that, I would like to thank you and happy to take questions.